you know, it's customary when people give medical talks to give references. And it's, all I can say is I'm going to give you places to, references to read where all these things that I say can be found. But I'm not going to take the time to cite literature studies during the talk. It's just too distracting, and I don't think it's necessary. So the places to look, and in particular, if there's any medical professionals or healthcare professionals out there who want to check on things, I would strongly advise you to check out these sources that I'm going to say, particularly before you ask me any further questions. There's just so many questions now that I would really rather you check these out. You can find all the references, all the citations that you need in these. And the first one is a book by a fellow that I met named Klaus Kohnlein, K-O-H-N-L-E-I-N, and Torsten Engelbrecht. And the book is called Virus Mania. That's Virus Mania by Klaus Kohnlein. The second is a website by that's called theinfectiousmyth.com, and there's a lot of good references, particularly about the current situation in there. The third one is a book called The Silent Revolution in Cancer and AIDS Medicine by a German named Heinrich Kremer, K-R-E-M-E-R. And there's a lot of citations about medical therapy and viral therapy in there, so everybody should take a look at that. The next one is the book called The Invisible Rainbow by Arthur Furstenberg, and that's specifically talking about the health consequences of electromagnetic fields. And I got a lot of questions about the reference for when I mentioned that about what Rudolf Steiner said about viruses, and that quote came from a lecture series called Cosmic Working of Earth and Man. So that's Cosmic Workings of Earth and Man. And then later on, I'll refer to a talk that was given at the NIH. They have an annual talk to update essentially the NIH about the state of virology. This talk was given at the NIH on June 4th, 2015. The doctor who gave it was named Skip Virgin, and the title of the talk was called, quote, The Mammalian Virome in Genetic Analysis of Health and Disease Pathogenesis. And I can tell you it's not an easy listen. It's very technical, but for anybody who's really interested in going deeper into that subject, I would say you really must listen to that. The next introductory thing I'm going to say is a number of years ago, I wrote a book called Human Heart, Cosmic Heart. And that's our website, humanheartcosmicheart.com. And in there, I made what I thought was controversial statement at the time, saying that, in my opinion, the heart does not pump the blood. Now, I would just point out that right now there's two people around in the country, in the United States anyways, saying the heart does not pump the blood. There's another reason why the blood moves in the body. There's me and an anesthesiologist named Bronco First. And he wrote a book called The Heart and Circulation. And the foreword to that book was written by the head of cardiac anesthesiology at Harvard Medical School, who basically said Bronco is correct and it's not possible that the heart pumps the blood. And so as I've often said, I may be wrong, but apparently at least I'm not the only one wrong about this. And the reason I bring that up right now is in the front of that book, I wrote a quote from a guy named Victor Schauberger, who many think, including me, probably knows more about water than any human being who ever lived. And I'm going to read that quote because I thought it was appropriate for the Heart book, and I have a feeling it's even more appropriate for right now. So this is a quote from Victor Schauberger. Quote, People may say I am crazy. Perhaps they are right. In this case, it is not so much important if there is one fool more or less in the world. But in case that I am right and science is wrong, Lord, have mercy on mankind. So I just want to start with that as a way to say, obviously, this is my opinion. This is my take on the situation. And everybody is free to make of it what they want and investigate it further. And I hope that people do that. So the place I'm going to start here is we have this situation in the world where apparently there's a lot of people getting sick, and it started in a place called Wuhan, China. And now there was some other main places like in northern Italy, and then essentially all around the world, or at least a lot of places in the world, there's a lot of people getting sick. So the question is, why are they getting sick? And of course, the usual theory that we're all being told is that there is this new sort of novel virus called a coronavirus, and that is the cause of why everybody is getting sick. And I thought that the first step here is we should look at the history of how we know that a certain disease or a certain set of symptoms is caused by a microorganism at all, and then specifically by a virus, because that's really the place to start. And so the way that I would explain that, and this is basically formulated in a set of postulates that started in the essentially the early part of the last century, and they're called Koch's postulates. And these are the accepted way we prove that any set of symptoms is caused by an infectious agent. And they're used and essentially approved by all the infectious disease people and virologists in the world. So Koch's postulates are basically four. And let's take a very simple example so that everybody can understand this. The first postulate is that people have essentially the same set of symptoms. And so let's take the example of meningococcal meningitis. That's a type of meningitis that's caused by a bacteria called meningococcus. And essentially, all the people with that meningococcal meningitis, they have a high fever, they're extremely sick, they have a very characteristic rash, and if nothing is done, and even sometimes if anything is done, they will die in 2 to 3 days. And that's 100% of the people have the identical symptoms. Now, once you have a person with those symptoms, you can take some of their blood and you can do what we call a blood culture, where we attempt to grow an organism out of their blood. And what you find is 100% of those people with meningococcal meningitis grow out a culture of pure meningococcus in their blood. 100%, not 50%, not 80%, 100%. The next thing, and that's postulate number two. Postulate number one is people have more or less the same symptoms. Postulate number two is that in those people with the symptoms, they all have the same organism, and only that organism growing out in their blood. Postulate number three is that none of the people walking around who are normal, presumably like most of us, none of us have meningococcus growing in our blood. It's simply not compatible with normal health. So that's postulate number three. Postulate number four is once you have a pure culture of meningococcus and you grow it out, you can isolate and purify that bacteria. You can then inject it or expose another person, which you wouldn't want to do, or an animal who's susceptible to that bacteria. And 100% of them will get meningococcal disease. And then 100% of those, you can then grow out meningococcus in their blood. And then you could keep that thing going basically forever. Now, let me just stop here for a minute because I realize that this talk is going to get very technical for some. And frankly, I just don't know any other way to do it because this is the things that I think you need to know. So in other words, to summarize, the way we prove infectious etiology or infectious causation is we have people with the same or very similar symptoms. We grow out a pure culture of that organism, whatever it is that's causing it, from the blood or the respiratory secretions, and only that organism is found. Then we take that organism, we purify it, we inject it into a susceptible animal, and they get the same disease, and then we culture that organism out from them. And that proves that it's that organism caused that disease. Now, you can do the same thing with viruses. So let's take an example of chickenpox. It's a common childhood viral infection. More or less, every child with chickenpox has the same set of symptoms. They have a low-grade fever, sometimes it's a little higher. They don't feel so good. They have a vesicular or bubbly rash, and that's how we know it's chickenpox. Now, you can then take the blood or the fluid from these bubbles, and you can look at it under an electron microscope, and you will find millions of copies of the virus in the blood. That's called viremia. Or you can find thousands, maybe millions of copies from the bubbles, the blisters on their skin. You don't find any other virus. You find a pure culture of this chickenpox virus from either the blood or the vesicles. You can then look at the at normal people. Sorry, I misspoke about third postulate with meningococcus is no normal people have meningococcus growing in the blood. Similarly with chickenpox, there's no normal people like you or I walking around who have millions of copies of chickenpox virus in our blood, and we don't have any of these vesicles. And if we do have had a blister, it wouldn't have any chickenpox virus in it. And then number four, the fourth postulate, is you could take some of this virus, you could purify it. And when I say purify it, I mean you do something to it, filter it or whatever, so that the only thing you're injecting or exposing the animal to is a pure culture of chickenpox virus. And then 100% of them who are not immune, they will get sick, they will have chickenpox virus in their blood and then their blisters, and then so on. That is how all doctors, all virologists, all infectious disease people understand how to prove an infectious origin. Now, there's a long history of this, which I'm just going to briefly mention, because when you look at what's happening in with this so-called coronavirus epidemic, the first thing you notice is this started in Wuhan, we're told. And then you say, what were the symptoms of the people in Wuhan? So the best that we know of, the symptoms were low-grade fever and dry cough. Now, unfortunately, or maybe not unfortunately, but realistically, that is not a very specific set of symptoms. Lots of people have low-grade fever and dry cough, including people who have bronchitis, including people sometimes with asthma, people with exposed to air pollution, and a whole host of other things. So that first postulate, everybody has the same symptoms, is actually not met in this situation. And so then the next thing is, do all those people, okay, so maybe they all have a low-grade fever and dry cough, and that's just what it is. What should have happened, and what, if I was the CDC expert and I was called to investigate, or if you call the Chinese virology authorities or the American virology authorities, if you say it looks like a new disease, there's people who have a dry cough and low-grade fever and the shadowing on their CT scans of their lungs, we've never seen that before, can you come and investigate to to see if there's an infectious etiology. Okay, fair enough. So what I want you to do is line up 500 people, all with that same symptoms, and we're going to do an electron microscopy examination of their sputum and their blood, and we're going to find out if all of them have millions of copies of this, of a virus, and then we're going to characterize the virus and see what happens. The next thing I would do is I would ask them to take 500 people who are perfectly normal, because you need a control in this, and say, let's look at their blood and their sputum and see how many of them have millions of copies of the virus in their blood or their respiratory secretions. And the answer should be 100% of the first 500 have millions of copies, and 0 of the second group have this millions of copies. Then we would purify those millions of copies of the virus from the first group, find a susceptible animal and either inject it or put it in their nose or squirt it down their throat. We would do then a control, so we would take a saline solution or something like that and do the same thing, except it didn't have the virus. And if all of them got sick and all of them had millions of copies, we would prove that this was because of this new virus. So that's how this, quote, should have been done. Now let's look at how it was done. As far as we know, and where I'm getting this from is a search for PubMed, a search for articles, and people are sending me things to corroborate this, and so I would go to infectiousmyth.com. As far as we know, not only are there not 500 people who had this done, as far as I can tell, there was nobody who had this done. There was not a single person who had these symptoms who had millions of copies of coronavirus isolated from their blood or sputum. What they said was, and what they published, and I've seen the articles, is we have people who are sick. Here's a smear of their electron microscopy examination. And you see lots of things. You see cellular debris. You see a number of different things that look like viruses. And one of them looks like a coronavirus. So that's a completely different story than what I described for meningococcus or chickenpox. So, and then they did not do an examination, electron microscope, of 500 normal people. They simply didn't do it. Now, the people who wrote the articles then said, and I quote, "We did not attempt to purify the virus," which, if you think about it, is mind-boggling. Because if they didn't purify the virus, how do they know that that was the cause of the disease? And the answer is there's no way to know that. But they did take the virus and they did a genetic sequence of it. The genetic material of a coronavirus is RNA, not DNA. And they said, this is a new genetic sequence which we haven't seen in a coronavirus before. Therefore, this is the cause of the disease. And all I can say is this flies in the face of everything we know about virology and infectious causation. And I would also point out that this is not the first time this has happened. In fact, it happens basically every time. And let me just give you one example. Around the later part of the 1800s, there was people showing up with a new set of symptoms of acute paralysis. And eventually that was called polio. And because this was the time of Pasteur and the whole inauguration of the germ theory, by the way, as many of you probably know, apparently Pasteur recanted the germ theory and eventually said the germ is nothing, it's all the terrain or the experience of the person. But let's skip that for a minute. So they had these bunch of people with the symptoms of acute paralysis. That's what we call polio. It also was the time when they were first spraying the orchards and the apple trees with something called lead arsenic. Now, the part of your body that has the pathology, the part that's sick, is called the anterior horn cells of the nervous system. That's why you get paralysis. It turns out that lead arsenic and then later DDT are very specific poisons for exactly that part of the nervous system. So anyways, they said, well, this must be an infection. So what they did, and I would say what they did, believe it or not, but again, if you go to the Virus Mania book, you will see the entire documentation of this, is they took children who were paralyzed and some who had died, and they extracted some of their spinal cord that was diseased or their brain tissue after they died. They basically chopped it up like in a blender. They didn't purify it. They didn't see a virus. They didn't have electron microscopes then, so they couldn't see a virus. They didn't see a bacteria. They didn't even filter it. They just took this chopped up, essentially spinal cord tissue and gave it to monkeys to drink. But interestingly, nothing happened. And that was one of the big conundrums of this polio virus situation, is they couldn't find an animal that they could make sick. So then they injected it subcutaneously, like we could give a vaccine. And they injected it subcutaneously into the arms of monkeys, and none of them got sick. So then they decided, we have to prove that this is an infectious disease. So they took a child who was paralyzed. They took some of the diseased anterior horn cells from the nervous system. They chopped it up, took 2 monkeys, drilled holes in their skulls, and injected about 1/4 cup of unpurified spinal fluid goop into their brains. One of the monkeys died. The other one became paralyzed. You can see a picture of the researcher who was crowned as one of the shining lights of infectious disease, holding up the monkey, saying, this paralyzed monkey proves that it's an infectious disease. Now, I would say, if you ask me what to make of that study, I would say that if I was a monkey and somebody came at me with a bunch of goop from somebody who just died or had a disease in their spinal cord and said, I want to drill a hole in your brain and inject it in, if I was the monkey, I would run away as fast as I could, because clearly that proves nothing. Now, this same thing was, the same failure of evidence was done with the H1N1 situation. It was done with the H5N1 situation. It was done with SARS. It was done with Hep C. It was done with Ebola virus. It was done with Zika virus. This is not the first rodeo for the failure of the infectious disease community to follow the actual laws and rules that they set up to prove infectious etiology. So that's the first thing that I think everybody must understand. Now, a lot of you and a lot of people ask me the question, well, but don't we have a test that shows that this is caused by this virus? Now, sometimes, because I have a little bit of a smart alecky tendency, I must admit, I ask the people, do you know what the test was? Or do you know what the test is? So far, essentially, nobody has known what the test is. The test is called a RT-PCR test. It's otherwise known as a viral load test. And the test is a surrogate test. It was developed by a guy named Kary Mullis, who was given the Nobel Prize in Chemistry for essentially inventing the technique of this test. And he said very specifically, you cannot use this test to either prove infectious etiology or to diagnose an infectious disease, which of course is interesting, because if you can't use it to diagnose an infectious disease, that of course begs the question of what can you use it for? But let me back up here and describe what a surrogate test means, because this is very important to understanding the situation we're currently in. A surrogate test means that in a situation where you're trying to prove causation, you have to have a gold standard test. And those postulates, like with meningococcus, That is a gold standard test. It's reliable 100% of the time. And as some people know, I like to speak in analogies here. So here's a way to understand this. Let's say you want to go to a town and you want to know how many feet there are in that town. And I don't mean inches and meters, I mean the feet at the bottom of your legs, right? So the question is, how many feet are there in Podunk, Alaska? So obviously the gold standard test for that is to go to the town, have everybody come to the town square, and then count their feet. So then you have 1,112 feet, and you know that that's accurate. And then you go to the next town, and you say, okay, everybody come to the center, 100% of the people come, you count their feet, and there's 3,240 feet. You do that 3 or 4 times, and you've now established a 100% foolproof way of knowing how many feet there are in that town. Now, in that situation, you can use a surrogate test. And so what do I mean by a surrogate test? Then you say, well, I don't want to count everybody's feet. I know that everybody who has a foot has a shoe, and everybody who has a shoe has a shoelace, and there's only one shoe store in town that sells shoelaces. So I'm going to go to that shoe store and see how many people bought shoelaces this year, and that's going to tell me how many feet there are. That's what's called a surrogate test. Now, the problem with a surrogate test is every one of those assumptions better be correct. So for instance, how do you know everybody has bought shoes? Because maybe somebody doesn't like shoes. And then how do you know that everybody only has 1 pair of shoes? I've heard there's women who like 4 pairs or 6 pairs. And what about if some of the shoes, like I have a pair of shoes that doesn't have shoelaces? So what happens with that? And the point of that is, unless you know all of those things, and unless you have a gold standard to which to compare it to, you cannot use a surrogate test to prove anything. And that is what is happening with these tests. So what is the surrogate test? So remember that we don't have a gold standard. We don't have isolation, purification, reinfection. We don't have viremia. We don't have millions of copies demonstrated on an electron microscope. We essentially have no idea who has this coronavirus disease. So then they take a piece of one of the coronaviruses, the new one that they found. It has a new RNA sequence that hasn't been found before. They take one of the sequences, which they say is unique to that particular virus, and they do something called amplify it. And what that means is you take in your blood, you'll have 1 copy of this sequence, and it's too small, you can't find it. So you stimulate it, and this is what Kary Mullis came up with. You stimulate it, it makes 2 copies, that's 1 cycle. You make 4 copies, that's 2. You make 2 to the 20th copies, whatever number that is, that's 20 cycles. And what you find with this test is that once you put it through approximately 36 cycles, then you start to see the color change that tells you it's positive. So if you do 35 cycles, it's still too small to see. If you do 36, you start to see it, but you get false negatives, even though you don't really know which is a false negative because you don't have anything to compare it with. So then you do 37 and you see it, 5% of the time of people with these symptoms, and you say that's the number. But here's where it gets interesting. If you do it 40 times, you start seeing a lot more positives. And then here's something else to know. If you do it 60 times, so if you amplify it over and over and over again, it becomes positive with 100% of the people. Let me say that again. If you amplify it 60 times, it will be positive with everybody. That means that everybody has a piece of this RNA somewhere in their cells or in their genome or somewhere in their secretions. All you have to do is amplify it enough. And the problem is we don't know how many false positives or false negatives there are because we have nothing to compare it to. And if all biological tests have false positives. So if you test 30 million people and you have a 1% false positive rate, then 300,000 people by definition will test positive, and then you have an epidemic. And then if you want to demonstrate that the epidemic got better, all you have to do is lower the amplification cycles to 35, and then suddenly your, vitamin C or your vaccine or your chloroquine or whatever you did worked, and now there's no more people testing positive. That is fraught with problems, and that is the problem. And unfortunately, with every country has their own standard of what kind of cycles they put it through, the number. So you see very different numbers coming out of very different places, depending on the number of amplification cycles that they're standardizing their test to. And I hope everybody is getting the point that this is a crazy situation.